Genetic engineering. Genetic engineering is the process of altering an organism's genetic material, or DNA, to introduce new or modified traits. This can be done using a variety of techniques, including the insertion of new genes, the deletion of existing genes, or the modification of gene expression. One of the most common methods of genetic engineering is the use of recombinant DNA technology. This involves isolating a gene of interest from one organism and inserting it into the genome of another organism. The resulting organism, known as a transgenic organism, will express the gene of interest and exhibit the desired trait. Genetic engineering has numerous applications, ranging from medicine to agriculture to biotechnology. In medicine, genetic engineering has the potential to cure genetic diseases, such as cystic fibrosis and sickle cell anemia, by replacing or repairing faulty genes. Gene therapy, which involves the introduction of new genes into a patient's cells to treat a disease, is a promising area of research in this field. In agriculture, genetic engineering has already been used to develop crops that are resistant to pests and herbicides, as well as crops that have improved nutritional profiles. These modified crops have the potential to increase yields and reduce the use of harmful pesticides and herbicides. Genetic engineering has also been used to produce livestock with improved traits, such as increased milk production or disease resistance. In biotechnology, genetic engineering has been used to produce a wide range of products, including pharmaceuticals, enzymes, and industrial chemicals. For example, genetically engineered bacteria can be used to produce insulin, which is used to treat diabetes, or to produce enzymes that are used in the production of laundry detergent. However, genetic engineering also raises important ethical and safety concerns. There is the potential for unintended consequences, such as the spread of modified genes into wild populations or the creation of new and unknown diseases. Additionally, there are concerns about the ethical implications of creating genetically modified organisms, as well as the potential for discrimination or inequality based on genetic traits. As such, genetic engineering is highly regulated in many countries. In the United States, for example, the USDA and the FDA have strict guidelines for the approval and use of genetically modified organisms in food and agriculture. In addition, there are ongoing debates about the appropriate use and regulation of genetic engineering technologies. One of the most promising applications of genetic engineering is in the field of synthetic biology. Synthetic biology involves the design and construction of new biological systems or organisms with specific functions. This can be done using genetic engineering techniques to create custom DNA sequences that can be inserted into cells or organisms to create new functions or behaviors. For example, scientists are working on using synthetic biology to create bacteria that can produce biofuels or to engineer cells that can be used to target and destroy cancerous cells. Synthetic biology has the potential to create entirely new organisms or systems that could revolutionize fields such as medicine and energy production. In conclusion, genetic engineering is a powerful technology that has the potential to revolutionize numerous fields, including medicine, agriculture, and biotechnology. However, it also raises important ethical and safety concerns, and its use must be carefully regulated and monitored to ensure that it is used in a safe and ethical manner. With continued research and development, genetic engineering has the potential to create new and innovative solutions to some of the world's most pressing challenges. Genetic engineering also involves the use of gene editing technologies, such as CRISPR, Cas9, which allow for precise and targeted modifications to DNA. This technology has the potential to revolutionize fields such as medicine and agriculture by allowing scientists to selectively edit genes that are associated with diseases or crop traits. In medicine, CRISPR, Cas9 is being investigated as a potential treatment for genetic diseases, such as Huntington's disease and Duchenne muscular dystrophy. This technology could also be used to edit immune cells, allowing them to more effectively target cancer cells. In agriculture, CRISPR, Cas9 has been used to create crops that are resistant to diseases or have improved yields. For example, scientists have used this technology to develop wheat varieties that are resistant to a devastating fungus called powdery mildew. Another area of research in genetic engineering is the development of gene drives, which are genetic systems that allow for the rapid spread of a particular gene through a population. Gene drives have the potential to be used to control or eliminate disease vectors, such as mosquitoes that carry malaria or dengue fever. However, there are also concerns about the potential unintended consequences of using gene drives, such as the impact on non-target species or the potential for the spread of harmful mutations. In addition to the technical and ethical considerations surrounding genetic engineering, there are also important social and political considerations. For example, there are concerns about the potential for genetic engineering to exacerbate existing inequalities, such as by creating genetically modified organisms that are only available to wealthy individuals or countries. There are also concerns about the impact of genetic engineering on traditional agricultural practices and the environment. Overall, genetic engineering is a rapidly evolving field with enormous potential for both positive and negative outcomes. It will be important for scientists, policymakers, and the public to engage in ongoing dialogue and debate about the appropriate uses and regulation of genetic engineering technologies in order to ensure that they are used in a safe and ethical manner.